Okay, good evening, everybody. My name is Dr. Elizabeth Kramer from the Sydney Southeast Asia Centre. I'd like to welcome you to today's presentation from Dr. Justin Beardsley. Just before we get started, I'd like to acknowledge the traditional custodians of the land on which I am at the moment, and the University of Sydney. That's the Gadigal people of the Eora Nation, whose land the University of Sydney has been built on. And I'd like to pay my respects and the respects of the Sydney Southeast Asia Centre to those traditional custodians and acknowledge the ongoing tradition of education that's taken place on this land for many millennia. As I mentioned, a warm welcome to today's webinar with Dr. Justin Beardsley. This is part of our health webinar series that SEAC is running at the moment. Justin Beardsley has kindly offered some of his time this afternoon to talk to us about his research. Dr. Justin Beardsley is with the Faculty of Medicine and Health here at the University of Sydney, and he is a senior lecturer in infectious diseases. He's got extensive experience working in Vietnam, having worked there as a clinical researcher for several years. I'm going to hand over to Justin now. He'll be speaking for half an hour or so. The Q&A function is open for you, so please feel free to pop your questions in there and we'll get around to answering them at the end of the presentation with Justin.
Thank you very much for the introduction, and thank you everyone for joining online. It's always a great pleasure to get an opportunity to talk about my research and my interests, and I hope to share some of that interest with you and get some more people interested in the problem of antifungal resistance. Before we go any further, I just wanted to run a couple of quick polls. So to help me to get to know a little bit more about you as an audience and our participants, so I can hopefully change the talk slightly as we go through to address areas of your particular interest. So those two questions are related to the SEAC themes of research. So you can see those listed on your screen. Just tell me which is closest to your principal interest. And the second question comes down to fungi, because people have really different ideas when they think of fungi. It means different things to different people. And just choose the one that is most like what you think when you think about fungi. Is it the mushrooms growing in the park, or is it some kind of infection? Just let me have your thoughts on those. We'll probably wait 30 or 40 seconds to give you a chance to submit your responses. It shouldn't take too long, and then we can have a look at the results and hopefully tailor things a little bit as we go through the rest of the talk. Okay, do we have some answers there? Okay, so environment and resources is the most well-represented SEAC research theme, and mushrooms is what people think about when they think of fungi. So I'm not surprised that the mushroom is the fruiting body of the fungus and it's the most visible thing. It's not my personal interest, I'm afraid. So I hope nobody was really looking forward to talk about mushrooms. So I'm interested in the infections that happen in humans as a result of fungi, and those are infections caused by yeasts, infections caused by molds. And the one that people most often think about, but surprisingly nobody has clicked here, is thrush. And that's certainly the commonest infection. There are really serious infections caused by fungi as well. So those are the ones that I'm interested in and that we'll discuss a bit today. So this beautiful picture that you have on your screen is a rice paddy in Vietnam with those always tricky to navigate paths through them. And that's a little bit how I think about the problem of antifungal use in agriculture and human health consequences. So agricultural azoles are really important for food security. They protect crops, they protect produce after it's been harvested, and they hugely boost productivity by over 30% in some crops. And now, these days, current antifungals, primarily azoles, which I'll talk a lot about today, they're cheap and they're not toxic and they're very light. So you can treat a hectare of your farm with just a small 100 milliliter bottle in a spray can. That's worlds away from what people had to do 50 years ago. But it shouldn't be forgotten that fungal pathogens are really important in human health. They're kind of neglected killers. And although most people don't list, if I asked you about fungi, you list mushrooms at the top of the list, you wouldn't probably think of them as being really serious infections like malaria or tuberculosis, for example. But globally, actually cause more deaths than malaria and a similar amount to TB. And despite that, they don't attract very much research funding. So the estimates have been done exactly about the proportions of funding that are given to different diseases. And consistently, fungal research receives less funding than other areas. So people know less about them and are less interested. And in my mind, there's a mistake. Fungal pathogens have been responsible for extinction level events in plants and in animals. So there used to be a type of banana that everybody ate up until the 50s, but that's actually been completely wiped out by a fungal infection called Fusarium wilt. That's pretty astonishing. It's spread around the globe in a short period of time, so that the most consumed species of banana just no longer exists in the world. And a bit closer to home, or closer to the kind of human kingdom, we see an infection called chytrid, which is a fungal infection of frogs or amphibians. And over the last 10 to 15 years, that's also spread globally, and it's caused the extinction of more than 200 species of frogs. It's a devastating disease, and this horrible picture there is from somebody who's out calculating the loss amongst amphibians, and that's just hundreds of dead amphibians. So fungi are really important and under-recognized. And the thing about fungi, or particularly when it comes to human fungal infections, is we're very limited in what we can actually do about them. There's only 4 classes of antifungal drugs, and that's compared to 20 or more for antibiotics that would treat bacteria. So the armamentarium is really limited. And it's not just bacteria that get resistance, fungi get resistance too, but you can imagine if there's only 4 treatment options to begin with, resistance is a really, really big deal. And resistance comes about in several ways. It's always an evolutionary adaptation. So organisms will adapt to survive whatever toxins we put in their path. And if that toxin happens to be an antifungal, they're going to evolve over time to overcome it. And they can do that as a community by changing the species distribution. So species that are intrinsically resistant to antifungals may just become more predominant so that they can continue to spread. And individual species can develop resistance as an evolutionary mechanism. So they deliver mechanisms to resist the effect of the antifungal. And those 2 things have happened in the principal human pathogens. So that's Candida, which is the most important yeast, and Aspergillus, the most important mold. In Candida, resistance has come around mainly as changes in species distribution. So historically, the main species that causes disease in humans has been Candida albicans, and Candida albicans is pretty reliably susceptible to antifungal drugs. But over recent decades, we see different species, so Candida krusei, Candida tropicalis, and other intrinsically resistant friends are becoming more common in human infections. So they're redeploying the warriors that can overcome our antifungal defenses. And in fact, a whole new species has emerged in Candida that got some unusual media interest for fungal infections. So that was called Candida auris. And that is a kind of a global phenomenon that started around 10 or 12 years ago. It appeared all over the world, and it's resistant to many drugs, it's extremely tenacious, and it's made the New York Times because when it gets into a hospital or an intensive care unit, for example, they have to close it down. It's very tenacious, difficult to get rid of, and it's causing almost untreatable infections in the patients who are already very vulnerable. So it is a big deal. In Aspergillus, it's slightly different. So the main mechanism for resistance that we've observed so far is that main species, Aspergillus fumigatus, used to be susceptible to all antifungals, but is progressively acquiring mechanisms of resistance. And the picture there is, it's from I can't remember the magazine, but it's a really nice picture where they covered this issue. And that's killer tulips with spores, with these deadly fungal spores attached to them. And the reason that we have tulips is because this was identified first in the Netherlands, where they have a very big tulip industry, and they linked the emergence of resistance to that tulip industry. So if you are growing tulips in the Netherlands, you have the bulbs, and that's what's really valuable and that you want to sell around the world. And you definitely don't want those bulbs to rot after you've harvested them. So you treat the bulb with an antifungal before you store it. So antifungals are really heavily used in the tulip industry, and all the waste produce is put into a big compost heap. And compost heaps are basically where Aspergillus is born to exist. That's what it does environmentally. It breaks down organic matter. And sorry, slide just changed itself. And so it's working in the compost heap, but it's exposed to these azoles, and evolutionary, it's inevitable it's going to develop resistance. And so this problem was first picked up in the Netherlands, where they found resistance rates, surprise resistance rates of 10 to 15%. Subsequently, it's been described in lots of different settings, but not so much outside Europe and North America. So it hadn't really been widely described in Southeast Asia, for example. But we do know that the resistant organisms infecting humans are caused by the environmental contamination with agricultural azoles. And as Dr. Kramer introduced, I used to work, and my clinical research was in Vietnam for 5 or 6 years. And so I have an interest in Vietnam. But Vietnam is also a really good place to start looking at this problem of antifungal resistance. It's got a high burden of disease. They use agricultural azoles really extensively, and it's unregulated. So if you're a farmer in Vietnam and you think your crop's not doing well, you just go to the chemical shop or get the chemical agent to come and you buy these cheap drugs and you apply them to your crop. And the final thing is that there's actually currently no surveillance of fungal infections in Vietnam. So even if resistance was emerging as an issue, we wouldn't necessarily know about it. So for these reasons, I wanted to work in Vietnam. And I wanted to work in this One Health paradigm. So picture on the screen now is of the One Health triad, and that's showing the interrelatedness of human health, animal health, and environmental, soil health, agricultural practice, and how they're all interlinked. And particularly when it comes to something like fungi, who are depicted up in the top right of the diagram, because what happens in the environment has potential impacts on the health of humans and the health of animals and animal husbandry. And they all cross over and interlink, particularly for environmental organisms, which are fungi. So we decided what we wanted to do. I had colleagues from vet and agriculture and medical backgrounds, and we wanted to research the problem of agriculturally driven antifungal resistance in Vietnam. It's very difficult to get funding for this kind of research because it falls between different research funders, and people are not familiar necessarily with the methodologies that you propose. So we had to find a friendly funder. And we were really fortunate to be given some startup money from the University of Sydney's Sydney Institute of Agriculture. And the project that we proposed to them was looking at some sentinel organisms, so rice blast disease, Aspergillus fumigatus, and Candida albicans, and taking a One Health approach to looking at these different problems. And I'm going to talk to you a little bit about what we've done so far with that funding and where we're going to. Actually, as a result of the work around Aspergillus, I was given a NHMRC fellowship. So I'm going to be focusing on that for the next 5 years, and hopefully the other areas will be developing as well. Excuse me. So this is a picture of one of our early field trips with some research colleagues and of course just some local people, where we were going out to meet farmers. I wanted to start not in a really formal way, but just in an informal, friendly chit chat, talk to local rice farmers and fruit farmers about how they're using agricultural chemicals, what they understand about them, if they recognize there's any potential risks. And it was a great experience. We learned that they use chemicals really as growth stimulants. They're not really thinking about treating disease. They're just thinking about maximizing their crop return. And they're not making loads of money. So they're not like big chemical consumers making huge riches. You know, they're living on the edge and they need every little benefit that they can get. But we had lunch at one house and we ate some of their papaya. And I said, you know, they're papaya farmers. And I commented how delicious the papaya was and where would I have to go in Ho Chi Minh City to buy the papaya when I got home. And they kind of laughed and they told me that they don't eat the papaya that they farm because it's so covered in chemicals, and this was a papaya from their own garden. So even if they don't, they know on some level that the chemicals are not what they want. So through some of our results, first, rice blast. You can see here a picture of the devastation wrought by rice blast. It's a fungal infection. And we wanted to see if the way that agricultural azoles are being used is causing problems of resistance in that target organism. So remember, when they use agricultural azoles, they're trying to treat rice blast. They're not expecting to cause problems of resistance in Candida and Aspergillus. So this is really important to know whether they're actually using the right drug. So we traveled to 100 sites in central and southern Vietnam, where we collected samples of soil and diseased leaf, and we cultured the rice blast and we're now in the process of testing. So we haven't completed this project, but we're finding already quite high rates of resistance, which calls into question the way that the chemicals are being used in the first place. Not only are they having unintended consequences, they might not be having their intended consequence. For Candida, we focused on chickens. And the reason to focus on chickens is that azoles are really widely used in humans, as I've mentioned, but also in chickens. They mostly use it in animal husbandry, in chicken raising, to treat the sawdust, because sawdust can harbor molds that are deadly to chicken hatchlings. So that's the intended, that's the target that they have to suppress that mold. But a potential unintended effect is that chickens also carry Candida, and they carry the same Candidas that humans do. So the chicken constantly exposed to low levels of antifungals in their bedding may become colonized by resistant Candida, which could be passed on to humans, and which could be responsible for these changes in species distribution that we're observing, or even the emergence of new species. Now, this is just my hypothesis. This has not been proven in any way, but I think it's kind of exciting. So it's really looking at that interaction with the farmer and the farmer's chickens, then the meat, which is also kind of colonized with the Candida, and the person who's preparing the meat, and obviously stretching out to the end consumer, and whether the farm practices can spread all the way through that chain and cause problems to the end consumer. So one of the questions that hasn't yet been definitively answered is whether humans and chickens do share Candida. We know they have the same species. So Candida albicans can be found in a human just as easy as it can be found in a chicken. They're a natural part of our kind of microbiome. But we don't know that those are the same, exactly the same sort of lineages, I guess. It's possible that there are genetic barriers that would prevent it being transmitted between humans and animals. And people have looked at this, but they've kind of come down on both sides in terms of conclusions. Some people found that they're different. Some people found that they're essentially the same. And it's because of the techniques that have been used that are kind of fingerprint genetic tests rather than a whole genome sequencing approach. So in this project, we're collecting samples from chicken farmers and from the chickens, and we're going to use whole genome sequencing to look in much more detail about the relatedness and the potential for transmissibility. A couple of outcomes that we hope to find. One, if they're completely transmissible, then we would say there is a zoonotic potential in chickens. So that's a new discovery, and it's important because we would then have to think about occupational exposures, and we'd probably be more cautious about the way we're using drugs in chickens if we're conscious that this might have a direct impact on human health. Of course, it's pilot data, so it's to lay groundwork for the future and also for surveillance. So this project is in progress but not completed. Which brings us to our final pathogen for today, which is Aspergillus, and this is the project that's furthest advanced. Aspergillus causes a range of infections. It's the commonest mold. You'd see it on your bread. It's kind of fluffy. And we're breathing it all the time. Everybody breathes it all the time, but most of us don't get sick. We inhale the spores and we'd never develop disease. To develop disease, you pretty much need, or most people will have either a problem with their immune system or a problem with the physical structure of their lungs. lungs that have been damaged by previous infection, by smoking, by air pollution, or the commonest global cause, in fact, is TB, so it's a specific infection. The range of disease that you can get goes from a kind of inflammatory disease and an allergic disease, so you're just allergic to the spores and it can cause asthma or make your asthma very severe. That's at the kind of the lowest end of the severity spectrum, right up to people who've had anti-cancer chemotherapy can get a rapidly fatal infection called invasive aspergillosis. The disease that I'm interested in, and the disease that's most common in the whole, like in the global context, is chronic pulmonary aspergillosis. So it's Aspergillus infection in the lung that's just chronic. And the leading global risk factor for this is TB. And there are huge numbers. So if you had 10,000 people who've been treated for tuberculosis, there's 800 people who are likely to get infected with Aspergillus by 2 years. And of those, half will die. So 800 infections, 400 deaths. And these are not trivial numbers, especially in settings like Vietnam with high populations and the high incidence of tuberculosis. You can treat this infection, but there's a risk that if you're working and living in contaminated environments, you'll inhale spores that are intrinsically resistant to the first-line treatment, and your outcomes will be much worse. So that's depicted here in the azole-naive environment, where you'd inhale spores that are susceptible to treatment. So if you're unlucky enough to get infection, it can be treated with azole drugs. But if you're working and living in environments with lots of azole use in rice farming, shrimp farming, fruit farming, chicken farming, or whatever, you may be infected with a resistant organism that won't be susceptible to therapy. And these are the people that I'm working with and who I think this is the biggest problem for. So these are rice farmers in the Mekong Delta, have very high incidence of TB, and they're working and living in highly contaminated environments. And they don't have money for second-line expensive drugs. If the first-line ones don't work, then they're just going to be added to those statistics. Sorry, excuse me for 1 second. I'll take another drink of water. So I'll just share with you now some of the results from our research. What have we been doing to start looking into this problem with Aspergillus? We went to a very rural province of Vietnam called Ca Mau. It's way down in the south. It's a really farming province. It's agricultural, rural. It has some densely populated areas, but mostly it's pretty rural. And we chose districts that were predominantly rice farming, fruit farming, shrimp farming, national parking, and urban settings, which are the principal land uses down there. Within those areas, we selected random GPS locations. We went there with an air sampler and a shovel, and we collected air, we collected soil, and we sent it to Ho Chi Minh City, where our lab was based. And in the lab, I include these because it took ages to make these slides when I made them, and they're really pretty, but we don't need to go into too much detail. We basically cultured those things that we had on standard media with and without antifungal, and we looked at them through the microscope to identify which organisms they were. We stored them, we did various different approaches to culture, secondarily culture, harvested the Candida, and ultimately we tested susceptibility to a range of different antifungal drugs to get towards answering our question. So having shown you those things in cartoon format, I'm going to show you a really brief video that we made of our work. Myself and Chami, who's my postdoc based in Vietnam, and she's fantastic. I will forewarn you that the reason I have the picture of this lady playing the instrument is that the soundtrack to this video is quite unusual. It's music of danda, which is a kind of gigantic marimba made of stone. I'd asked my colleagues to identify some folk music that would be good backing for the kind of message that we wanted to get across. And well, I'll look forward to your opinions on it. I think the choice of music is quite interesting, but nonetheless, hopefully you will enjoy the video. You can see in real life what our work looked like. Okay, I'll be back in 3 minutes. Yes, it actually looks like the resistance that we're observing in Aspergillus has emerged as a result of the use of azoles in agriculture. Because there are so few drugs available to treat any kind of fungal infections, we actually use the same azole drugs on crops that we would plan to use in our patients when they get sick. And in areas where there's a lot of azole use, we find a lot more resistant Aspergillus in the environment, which increases the risk of our patients turning up with an infection that we can't treat with standard therapy. So it's a real concern. And no one's even begun to look at this problem in Southeast Asia.
In Vietnam, the agricultural chemical industry is fully regulated. Company agents sell azoles directly to smallholder farmers. Azoles are being used a lot in Vietnam, so it's likely that azole-resistant Aspergillus will be common. This, combined with Vietnam's high rate of tuberculosis, is a real concern. So this year, we started to sample the environment in Mekong Delta to better understand this risk. Our results so far show higher rates of triazole resistance than have been detected anywhere else in the world. The resistance rate we are seeing is more than 50%.
Our results so far, they really are of concern. And our next steps are to finalize those to really nail down the instance of resistance, and then to find out not only if this is a problem in the Mekong Delta, but is it a problem across the rest of Vietnam and the region. Ultimately, we need to work out how much of a risk these resistant bugs are presenting to the people that live in our region. Okay, I hope you enjoyed that short clip. Just to start then thinking about some of our results, what did we actually observe? I will show you some of our plates. So on the left is a standard kind of medium that we use to grow fungi that we collect in an air sample. And on the right is a plate that's actually got antifungals in it. So it shouldn't grow any colonies at all. Each of those things is one colony. And we spoke to our colleagues who've done similar research in other countries. They would do hundreds of samples, and on that antifungal plate, they would maybe grow one colony from a couple of locations. But basically, you should see nothing. This is actually relatively few colonies from what we observed. Basically, all of our samples, we were growing fungus on the antifungal treated plate. So we knew there was going to be a problem from the outset, and there was. So this chart, again, don't need to go into too much detail, but just be conscious that red is resistance and red is bad, and that we should be expecting to see rates of 10 to 15% for resistance maybe. And even in the columns on the left, which is National Park, our rates are up around 30% there. So twice what we'd expect, and that's in a pristine, uncontaminated environment, ranging up to much higher, 50, 60% or 70% in shrimp farming and urban residential areas. So really disturbing findings, which we need to investigate further. The green line that you see on that chart is where we found the antifungals actually contaminating the environment, what number of sites. Mostly you shouldn't be able to detect it because they don't last forever, and you shouldn't be using so much that it's just residually detectable in the soil. It should just be treating the plant and then gone. But we are finding in 20 and 30% of our sampling locations. Interestingly, we didn't see it in the shrimp farms in the urban residential areas where the highest resistance was. And we're still working on reasons for that. Shrimp farms probably make sense because there's a lot of water going through and frequent flooding and drainage. So it could be just washing it away. They're also very coastal, so affected by tidal flows. The urban residence is really interesting. You wouldn't expect the soil in an urban residential area to be treated with antifungals. So why do we see so much resistant Aspergillus? I'm not sure, but one possible explanation is that all the farm produce from around about is brought into the city to be sold. And so you have huge amount of vegetation. And once the vegetables are sold, for anyone who's been to Vietnam or anywhere in Southeast Asia, you'll know around markets, there's a lot of kind of rotting vegetable matter. And as I mentioned, that is the most favorite location for Aspergillus to hang out. And that's all contaminated farm produce with agricultural chemicals on it. So my working hypothesis is that that contaminated, composting, rotting vegetable matter that's hanging around the market is breeding resistant Aspergillus and getting exposure to like really dense urban populations. So it's a worry. So my final results slide here is comparing, just to give you a global context, the rates of resistance that we found in Vietnam compared to some other selected countries. I mean, more countries have done testing, but we don't see high resistance rates like we observe in Vietnam. Here, I've included Thailand as our kind of nearest neighbor, and they had 10% resistance rates. Some places, Indian tea farms, have observed resistance rates of around 30%. That's the highest I found in the literature. So really quite astonishing. So as I've pointed out, agriculture azoles are really important. They make a big difference to food sustainability, to people's ability to have affordable food, and to the incomes of farmers. But there are some definite risks to human health that we're picking up here in sentinel pathogens. And so it is a tricky balance, and I'm happy to discuss that further. Hope you've enjoyed the presentation. And if you have any questions, well, we'd be delighted to have them. So thank you.
Thank you so much for that, Justin. And I should have mentioned at the beginning of the before you started your presentation, congratulations on your NHMRC fellowship.
Thank you.
That's a really wonderful achievement and speaks to how important this research really is. I've got one question that's come through in the chat, which I'll get to in a second, but for all of the attendees, if you have any questions for Justin, now's the time. You can enter them into the Q&A function at the bottom of your Zoom screen, and I will get to them as they come through. But our first question here is from Daniel Howell. Hi, Daniel. Who asks, who is more at risk of antimicrobial resistance exposure to aerosols or pesticides? Would it be the people who consume the food or the rice, or the people who are spraying the pesticide themselves?
Yeah, thanks for the question. Well, I mean, it definitely varies by where those people spend most of their time. So I think typically it's found in the actual organism they're exposed to is either in their environment at work or in the home environment. And so as I saw in the chart about the different land use types, I actually think a bit surprisingly that the highest risk people are the people in urban environments, which is unfortunately where most of the people are. So that's a big problem. Inhaling the chemicals as they're sprayed is not a problem. It's inhaling the spores that are naturally produced by any decaying vegetable matter, be that in the soil or in the compost heaps around the soil, or in this rotting vegetable matter in the city. If any of that is contaminated with azoles and you inhale those spores, then that's the problem. Yeah.
Great, thanks, Justin. I've got a couple of questions. So interesting. I was sort of thinking about, what the implications of all of this are. You mentioned that there's no fungal surveillance happening in Vietnam at the moment on a wide scale. So if some kind of infection was to break out in one part of the country, what would that look like in terms of spreading across to other regions of Vietnam? How fast would that happen?
So a very good question. The spread of fungi is like really rapid and extremely widespread. So without going into sort of too much details about even yet new pathogens, we know that those spores, they can travel in the stratosphere. They're carried by winds, they're extremely light, they're dusty, and they can cross continents. There's no kind of limits on them. So that's why we think this is an important regional health threat. It's carried on vegetable matter that's exported, so it can be carried on tulip bulbs. It's carried on any transported vegetables, and it's carried just in the air. It doesn't, most fungal pathogens are not that important for being transmitted human to human. They're transmitted from the environment to susceptible humans, but they can go around the world really quickly.
So I guess that leads to a follow-up question that I had about whether there's any awareness or interest from the Vietnamese government in getting involved in this area, and what kind of interventions you're imagining they could assist with, or not just the government, anyone could assist with in order to prevent that outbreak from happening in the first place.
Yeah, thanks. Also, very important question. So from an official kind of angle in Vietnam, the biggest interest and supporter of our research at the moment is the head of the national TB program based in the National Lung Hospital in Hanoi. And the reason is because I mentioned about TB being the leading risk factor for developing these fungal infections. And you can, get your head around easily that if you're running a national TB program, you're spending your blood, sweat, and tears and your country's resources on curing tuberculosis. You don't want them to 6 months later get a new infection that looks very similar to tuberculosis and die. You want to know exactly what's happening. You want to know what's the best available treatment. And so we have Professor Nyum's support in investigating the resistance profiles and really understanding how many people are actually being affected and what's the best way to treat them. On a more kind of surveillance level, we had support from NIHE, which is the National Institute for Health and Epidemiology, also based in Hanoi, for a previous proposal that tragically wasn't funded, but that was intending to set up a network of surveillance labs, both in Vietnam, but also around the Mekong region. So I think there's interest, but like everything, it comes down to money. And if there was unlimited funding, there'd be, great interest.
Yeah, I mean, it's interesting that you say that, because it sounds like such a huge issue. You mentioned at the beginning of your talk that it affects just as many people as TB and kills more people than malaria. Do I have that correct?
Correct.
Yeah. So, I mean, in terms of the global interest in this issue, why haven't I heard about it? why haven't most people heard about it?
Yeah, I don't know. I've been banging this drum for a little while. Truly it is getting a higher profile over the last 10 years. I'm not sure why it didn't have a very significant profile before. Some of it is related to where the research funding goes. I don't know.
Yeah, I mean, obviously, if it was up to you, we would all know about it. And people who've dialed in here and who are watching on Facebook Live will know more about it by the end of this talk. But it just seems like such a blind spot for all of us. I mean, you did mention too, when you were talking to farmers, that they seem to have at least a latent awareness of the risks of using pesticides.
Mm-hmm.
Did you get a sense that they feel the same way towards the fungal treatments, or is that something that hasn't really, crossed minds, or where are they sitting in this picture?
Yeah. So Vietnamese farmers and are not unique at all compared to farmers in Cambodia, Laos, Thailand. They don't really distinguish between the different classes of chemical and the diseases that are being treated. They have a sick crop and they want a chemical to make it better, and they often just use a whole selection of different chemicals. So they use antibacterials, antifungals, and pesticides, traditional pesticides that are treating kind of insects, but they're all considered pesticides. So they have an awareness that they don't want the fruit that's got chemicals on it, but at the same time, they're very aware that productivity is 30% better if it does have chemicals on it. I've found in my experience, so in those kind of casual conversations, and also increasingly in the cities, Vietnamese people are into organic food. They like the idea of food that's not been treated by chemicals. So generally, there's a good understanding, but there aren't solutions and alternatives. And that's why it's so important that we're working with people from veterinary and agriculture and medical, because there's no good just saying, oh, look, there's a health threat from using azoles, because no one's going to give up their livelihood, and you have to provide alternative solutions.
Yeah, and that's really part of the puzzle, isn't it? You know, once you've figured out exactly what's going on, what's the solution and what's the next step? I've got a question here from Ruth. Okay, just give me a second to bring it up. Okay, Ruth's question is, better sanitation and hygiene is a big part of the solution for antibacterial resistance problems. Are you suggesting that it might also be part of the solution for antifungal resistance problems as well? In the Netherlands, with its horrid tulips, quote, there are wet markets, but market squares are cleaned at the end of the market day and there's no compost lying around. Could such an approach be of any use in Vietnam? And if it could be implemented?
Yeah. Yes, I'm sure that would be great. I mean, on the one hand, composting is is good, as a good use of the sort of leftover produce. The problem is that what you're composting is heavily contaminated with azoles. So, I mean, reducing the amount of contamination is really what the target should be. And there are physical ways, like sanitation, hygiene ways of reducing infection in your crops, for example. So, not planting so heavily, having airflow through your crops reduces the likelihood of getting the fungal infection in the first place. If farmers were better able to identify exactly what disease they have, they wouldn't use every chemical every time. So, you know, that could reduce the use of antifungals too. They just give an antibacterial when it's a bacterial problem, and they treat a pest if it's like an arthropod kind of pest. So I think those ways of reducing the contamination of the compost rather than just avoiding the composting altogether. But yeah, it would be nice if it was all scooped into one place and composted in a kind of municipal way. But still, those spores are not going to be are not going to be contained. I think there's no real way to contain them. They're so easily airborne that they will still cause exposure in the nearby populations.
Okay, thanks. And I've got another question from Ruth here. Do people who develop fungal disease as a result of lung damage due to TB or immunosuppression due to HIV, which you mentioned as predisposing factors, count towards TB and HIV mortality, or do they count towards fungal mortality?
So this is a really interesting question, and it's a big problem. They would count towards HIV mortality. They wouldn't count towards TB mortality. And TB mortality post completion of treatment is really poorly recorded. There's not much follow-up. And this is a big problem. So it's already been identified by researchers, Greg Fox, who's also based in the University of Sydney and worked through SEAC as well, that when you follow those TB patients who are cured, and several years later, they're still dying at a much quicker rate than people who never had TB in the first place. And the reasons for that are not clear at all. I mean, I suspect that one of the drivers is a fungal infection, surely, but that there are other things that we need to investigate. And I think a big problem in terms of frustrating TB surveillance is Aspergillus infection, because to all intents and purpose, sorry, a person with Aspergillus looks like a person with TB. They're coughing up blood, they're losing weight, they've got fevers. And in a really high TB setting, if they go back to the clinic and they were treated a year ago, the doctor's going to say, well, you've got recurrent TB, we should treat you again. And they'll do a test. But as we know, as you may know, people are often treated for TB, even though they don't have objective evidence of it. So it's called smear negative TB. And I think a lot of Aspergillus patients are treated for smear negative recurrent TB, and they die because they're getting the wrong therapy, and they're counted as TB related deaths. And so we're missing that really important information.
Just to follow up to that, is that part of the puzzle with making this more visible to the general public and maybe even to donors? I mean, if you could single out those numbers, would would that be helpful? And could you do it?
It's a great idea. That's my fellowship. So yeah, I mean, this was in the end how my project got funded. It is very difficult to get fungal research funded, to be honest. And this was how we were viewing the problem, presenting the problem. As this is an unexpected and poorly investigated complication of TB. It's frustrating TB surveillance, and it's worsening outcomes from what may be an effective TB program. And not only patients dying from a fungal infection unnecessarily, but they're being exposed to extra TB drugs, and that could be causing environmental contamination from a TB point of view and driving multi-drug resistance TB. So yeah, we're going to try and get those numbers, basically.
That sounds amazing. Well, so in terms of your research next steps, I mean, COVID-19 has sort of thrown a spanner in the works for a lot of people who do field work overseas. So how are you shaping up? How is the next phase of your project shaping up? What are you doing at the moment, and what are you going to be doing over the next couple of years?
Yeah. So I've been extraordinarily lucky that Chami, who we saw in the video, worked with me for a couple of years before COVID struck, and she's been able to carry on the work. So she was an early postdoc when she joined. She did a PhD in Seoul and was returning to Vietnam. And I think she's really, it's changed how we would have worked completely. Basically, before I'd have been there every 3 months for 2 or 3 weeks, we would save up visits to hospitals and directors and all that kind of stuff until I was in country. But obviously that was impossible. So Chami has really stepped up in a way that's quite difficult culturally in Vietnam for a young woman and a non-medical doctor to actually go and speak with hospital directors and TB program directors is really challenging. But she's really rose the challenge. It's been fantastic. So we've been able to keep things going and complete several projects. And in terms of getting ready for our next phase, we're doing a lot of Zoom, of course. Chami is going to Hanoi to build relations with our contacts at National Lung Hospital. We've established a research lab as a collaboration between University of Sydney and Hanoi Medical University, where we'll be able to do our more research-focused stuff. And hopefully, eventually, I'll be able to travel back and give her more support so she's not just having to do everything by herself.
Well, it is great to have that on-the-ground support already in place, isn't it? Especially prior to the beginning of last year. Just one final question before we wrap up. I'm just curious, and this is more of a general question. Working within Vietnam, how easy is it to do research there as a foreign researcher? And you know, are there any roadblocks that you've encountered for this particular research in setting it up? Or, you know, has it been very easy and sort of smooth? And if so, do you want to do a little plug for researchers refocusing on projects in Vietnam?
Yeah, I suggest everybody does research in Vietnam. I love working there. I think being there for 5 years and spending time getting to know how people work and the cultures and what is likely to be acceptable, understanding some of the slightly different mores when it comes to ethics approval, for example, just as a really basic thing. Ethics approvals are given by hospitals by and large. And if your research was answering a question or was asking a question that was very likely to result in saying the hospital was doing something wrong, that is never going to be approved in an ethics committee in Vietnam. And that's quite different to research in the UK or Australia or New Zealand, where I've worked before, where hospitals are often quite interested in knowing what it is they're doing wrong for purposes of improvement. So those questions just need to be phrased differently, and everything needs to be a win-win situation. I learned that. Learning some language. So, I mean, I've been learning Vietnamese for years and I'm still learning, and I've actually signed up for classes recently through Sydney Southeast Asia Centre. that helps a lot, just to be able to have some baseline conversations and let people know that you're committed to Vietnam and to working in Vietnam, and you have an interest in Vietnam and its people. I think with those things in place, it's not a very difficult place to do research. There's a great hunger and a thirst to answer questions. Very well-educated population with lots of people who've done PhDs and are eager to do more. And yeah, I think it's pretty good.
Well, that is a fantastic plug for anybody thinking about doing research in Vietnam. I'm going to end the session there. So thank you, Justin, for your time. To all of the participants on the line, if you want to share this video with any of your contacts who weren't able to make it, it is available on our Facebook page, the Sydney Southeast Asia Facebook page. It'll be up there as a live stream, but people can watch it after the fact. And I also want to give a quick plug for our upcoming event, which will be next Thursday, which is with Dr. Navneet Dad, and he will be talking about training animal disease detectives in the Asia Pacific to prevent the next pandemic, which I think is something that will be of interest to a lot of people, whether you are a veterinarian or a vet scientist or not. And on that note, thank you so much for joining me. Thank you everyone in virtual land, and hopefully catch you next Thursday at our next health seminar. Thank you.
Thank you.